Good morning, everyone. Thanks for being back here today. I'm Luca Foschini, co-founder and chief data scientist of Evidation Health. I'm really excited about this use case. It is actually part of the onboarding material at Evidation in my team. That's how excited I am about it. And it's a successful use case. It stemmed from observation of the patient need, and it went all the way to qualification as a secondary endpoint at EMA. And so it's great. I always bring it up as an example. The submission itself is just like 30 pages of so much information that could be used, I think, as a blueprint for other measures of qualification. So I hope you enjoy it. Without further ado, Laurent, oh no, actually Pat is the first one. Thank you.
Well, good morning, and thank you for including me. I'm happy to talk to you about Duchenne muscular dystrophy. Let's see. Here we go. So, lots of people have talked about Duchenne muscular dystrophy, and lots of people define it. I think that, for me, the best definition is Gower's, and it's the disease that's most interesting and at the same time most sad of all of those we study. For me, it's equal parts frustrating, fascinating, and heartbreaking. So when a person is diagnosed with Duchenne muscular dystrophy, here's what they learn. Those two little boys in the photograph are my sons, Christopher and Patrick. Christopher and Patrick were off their feet at 9 years old, and they were dead at 15 and 17. So today, what parents hear is that there are 79 exons, 2.4 million base pairs, which feels like a lot of foreign language when they hear those words. 60 to 70% of these children have deletions in that gene, but there are 1,000 deletions across this gigantic gene. So they learn that. They also learn that there are leaky genes, so an exon 3 to 7 mutation means that there's going to be some leakiness, and so that trajectory may not be as predicted, or exon 45. So there's a lot of complicating things that parents have to learn, in addition to the fact that they learn that this is a multi-system disease. So it affects skeletal muscle, cardiac muscle, it affects bone, it affects the endocrine system, it affects everything that these parents do, and they learn about their trajectory, which has dramatically changed over the years. So when my sons were here, the mean age of death was the teens, and they in fact were in that trajectory, dying at 15 and 17. Today, with the use of cardiac medicines and steroids and non-invasive ventilation, these boys are living longer lives. So the mean age of death right now, according to the Center for Disease Control, is about 29 years old. Again, but the trajectory of the loss of skeletal muscle function continues in much the same manner. These young men lose the ability to walk between 11 and 14 years old. They lose the ability to move their arms in their late teens or early teens, and then they're left with, as Jen said yesterday, being non-ambulatory. And in our trials, 6-minute walk test is what was the sort of primary outcome for those early trials. And you can imagine if you're on the verge of losing ambulation, or if you lost ambulation, it feels like you're pretty outside the bus and you don't have any hope. And so that was really unfair to those families, and certainly those boys upon whom all research was built on. It's also a complicated and expensive disease, and you can look at here at the requirements of this disease. Early on, we use steroids. For better, for worse, we're using steroids because they do protect muscle function, decrease inflammation, and they help these boys walk longer. Except that they don't grow, their bones are fragile, and they have all of the cushingoid features that you would expect with steroids. Also, when we look at trials, because of medical interpretation of how to use steroids, the start time, the dose, the regimen are all varied across this population, all varied across the United States and every other country. So as these boys increasingly lose mobility, what we see an increased expense and increased use of services, increased use of needs for these boys, and complicated medical care. So things are very complex in Duchenne, and they really, really deserve, all of them, to be in studies so that they can understand and learn. We can learn from them, but they can hopefully take advantage of what opportunities lie ahead of us in terms of clinical trials. I'm happy to say that since about the last 20 years, things have progressed, the disease is well characterized, and we have really possibilities to treat this disease, not only expressing and restoring dystrophin, but all of the downstream, inflammation, fibrosis, et cetera. So there are more than 42 companies in this space looking at how to modify disease progression, which is incredibly exciting except when you think about many of the trials. In fact, about 80% of the trials are ambulatory population, which reduces the amount of patients available for trials and reduces hope in those who've lost ambulation. So when we looked at this in terms of the 6-minute walk test early on, the first studies were all comers to 6-minute walk test. And forgive me for the men in the audience, but if you've ever tried to get a 5-year-old boy to concentrate for 6 minutes to do a walk, I mean, you're pretty good, because most of them are distracted, in addition to the fact that dystrophin is absent in the brain, and so they do have cognitive issues. Some of them have OCD issues, other behavior issues, and they're boys. And so trying to get them to do a 6-minute walk test is really interesting most of the time. And so that says, you know, again, how is it? Do you bring mom in and she cheers this boy on? We did a small study with Ohio State, or Nationwide Children's Hospital in Columbus, Ohio, and we wanted to see the difference if you paid them $100 at the end and said, do it again. We had a 100-meter difference in the 6-minute walk test if you bribed these little boys. So my own children did well with bribes, so we can see that as well. As we looked down at this and looked at more seriously at this, we found different cohorts. We found that boys who are walking above, 350, 400 meters are going to be pretty stable for a year. We found that boys below 200 meters could be predicted to go off their feet. So we learned that there are different groups within this group, and in fact, brothers are different. My son Patrick was off his feet at 9, and my son Christopher walked until he was 11. So what's the difference, and how can we really measure it in an appropriate way so that we understand what these drugs are doing and how to get them approved and get them accessible to these young boys? So I'm going to turn it over to Laurent.
Thank you, Patricia. So my name is Laurent Servais. I'm professor of pediatric neuromuscular disease at the University of Oxford. That's where my typical UK snooty accent comes from. But I come from Bath. Well, I'm not going to beat a dead horse, right? It's not the right place to explain you that a continuous measurement is much more powerful than a point assessment. I mean, it's quite obvious that if you can average the area under the curve rather than picking up a single point, you are much more powerful. Some addition about this 6-minute walking test. What we discovered using magnetoinertial technology is that patients train to enter into a trial. There is a kind of magic corridor, which is 300 to 450 meters, in which we believe that the patients have a linear evolution. And if the patient is not within this corridor, it cannot be included, which means that some parents are so committed to enter the child into a trial that they can train him to downgrade his performance, to go below 450, or to increase his performance, to be above 300 meters. So, which means that it's completely spoiled the trials and the statistic, because let's imagine that a patient walking 550 meters is trained to walk 450 meters at baseline. What happens if this guy is on placebo? He will improve naturally, right? So we discovered that some patients train, and this is also an important finding, is that when a patient leaves the hospital after the 6-minute walking test, he walks faster than during the 6-minute walking test because he's so happy to leave the doctor. Which means that when we record the 6-minute walking test, we record a kind of composite measure of a little bit of motivation, a little bit of power, a little bit of fatigue, a little bit of, we don't know what, and this is a point assessment. So it's what the patient is able to do that specific day. So the 6-minute walking test was a gold standard, and soon after was considered the fastest climbing test. And I think that it's so important one day, if you want to understand what these tests means, to come and to see these tests in the hospital. And sometimes I was discussing, with payers, and they were discussing about the 4-stage climbing test, and I told them, have you ever seen a 4-stage climbing test? Do you have the singlest idea of what it means? And I showed this video. Sorry, the video doesn't work? Is it possible to have the video? Oh. Okay. Okay, it's too bad. You could have been the physiotherapist and try to measure these patients during a 4-stage climbing test. The duration of the test is 2.3 seconds. Okay? So you have click, and then click. 2.3 seconds. And we are gambling decades of research and millions of investment into a test that lasts 2.3 seconds, and in which the time reflex of the physiotherapist is the main driving factor. So it's very important to come in the hospital and to see these stats. Come and see what is a scale and how the kids behave during a scale. Then you will stop believing in scales. Another point is what I call the Eiffel Tower syndrome. You know, I've been working in Paris for 10 years, which allows me to describe the Eiffel Tower syndrome. What does it mean? It means that basically the phase 3 trials are conducted in major cities, and this is our rare disease, so patients come from far away. So they are tired when they come to the center. And what is the first thing that a kid has to do when he comes to Paris? It's to go and see the Eiffel Tower, or to go to Disneyland. And I remember my first day at Institute of Myology, I say, what's happening here? There was a kid walking 6 minutes, and the mother told me, oh, he's not doing that well, you know. Yesterday we went to the Eiffel Tower, and he was exhausted. And it's the baseline, right? So after 50 trips in Paris, he doesn't go to the Eiffel Tower anymore. And it's going to be the last assessment. So that's why we started to think about a continuous measure, and we started like 10 years ago. It was 10 years ago I started working with Fitbit, or what was considered as a Fitbit at that time, because Fitbit came afterwards, and I would say the mainstream device. And the problem that we had was actually that this device were not calibrated, were not done to perform what I wanted to do as a clinical trialist, which was an alter of movement. I wanted to capture any single movement of my patients and to characterize any single movement, and you cannot do it with such device. You just cannot do it. And one of the reasons are the sensors, typically the sensors, the way they are calibrated and the way that they are sensitive to temperature. Because when the temperature changes, everything changes in a sensor. And all these sensors are calibrated for a temperature of 20 or 22 degrees. But when you go out the room and it's -3, then you comes back in the room, I can tell you that it makes a big difference in any measures that you take. How many of these sensors tell you what happened when you change the temperature of all of a sudden by 20 degrees? It's what happened when you go out of a room. So we didn't want to have this approach to say, okay, let's take a very beautiful device. We don't know exactly what it records. We give it to patients, and maybe we will have something out of that. So we wanted to really to start from the need. What do we need as physicians when we conduct clinical trials? And so we constructed this first prototype. It was in 2010, and we could identify what we needed in terms of precision and in terms of sensors. Then we had a second version, then a third version, then a fourth version that was a medical device, and finally we got this qualification from EMA 9 years later. So it was a long and winding road, okay? And nowadays we have a new design that is coming, and the concept was to at first to understand what we record, to understand the data, to understand what comes out of the sensor, and then to make something beautiful. So, first thing first, when we record volunteers, it's super. A stride is a stride of 90 centimeters is a stride of 90 centimeters. But when we record patients, we appreciated that if we measure the 6-minute walking test with a physiotherapist or with our device, we overestimated the 6-minute walking test all the time. And we were so stupid that we needed 2 weeks to understand why. And the reason is very simple, is that basically this is how it works, a 6-minute walking test. 2 cones from 25 meters, and the patients, they turn around the cone. But this is not taken into account by the physiotherapist. So which means that if you exactly measure the trajectory of the kids and you add this trajectory, then the correlation is perfect. Which means that at the end of the day, the real measure is not the one that makes the physiotherapist. And this is also true for the 4 stairs climbing test. Then comes the question of the variability. People will tell you, well, come on, you know, this is this is highly variable because it is a real world. And this is, you know, England, it rains all the time. So when it rains a lot, you don't walk. When it rains a little bit, you walk more. This is France. When the French are on strike, they walk more because there is no public transport. So two different kind of variability. And if we take the example of a kid who will live during 1 week with the mother, she's a sport woman, she exercises him, then he goes to the cousins, the mother's side, the parents are divorced, and they live in the countryside, and they walk a lot, and then he goes to the father, and he has no activity except barbecuing, and then he goes to the cousins, the father's side. So it's highly variable, but if you average on a 3 days, on a 4 days, 5 days, then you decrease the variability. And it's a very simple mathematical model. Basically, it's a bandpass filter, which means that any of the variable that you record from this kind of device, the variability of this variable will decrease with the time during which you average your signal. This is logic, and you can demonstrate it. And with the stride length, for instance, if you take any single stride from the patient, and you measure any single stride from the patient, and then you make a distribution, and you can be interested in the median, you can be interested in the 95th centile, this variability will decrease with the time, and when you reach 180 hours of recording, which is about 2 weeks, then it fails to about 3 to 5%, much less than what we consider as acceptable for the 6-minute walking test. Because for the 6-minute walking test, we accept a variability of 15%. So even at baseline, an assessment that is performed at home is less variable than an assessment that is performed in the hospital. Another point also is that counting meters will bring you into the wild. Counting strides, which you can do with a stupid podometer, will not help you, because yes, the variability is huge. It depends on the weather, it depends on the seasons, it depends on the father. But the stride length and the stride speed is much less dependent. If you characterize the movement, if you measure the movement, it is much less variable than if you simply count the strides or measure the activity. So why did we choose the 95th centile stride velocity? We just measured the standard response measure. So we looked for the centile of the distribution that was associated in the patients with the best standard response measure. And if you go up to the centile, you will see that the curve is the best at 95th centile, and then it increases because the patients are, because you then average a very limited number of strides. So this 95th centile stride velocity is basically the top performance of patients at home, right? The 6-minute walking test is the top performance of patients in the hospital, the most rapid walk in the hospital. The 95th centile stride velocity is the most rapid strides at home, when you aggregate the strides of the patients, without any kind of motivation, without the $100 of PAT, without the Eiffel Tower. You just take the patients and take the 5% most rapid strides. And it's basically about 6 minutes of walks, because if you record during 180 hours, a patient's walk about 30% of time, it's going to be about 5%, it's going to be about 6 minutes. So we could correlate this 95th centile stride velocity with different outcomes, the 6-minute walking test, the North Stars, the 4-stairs climbing test. This was correlated in patients, but not in control. And in controls, this was very discriminant between patients and controls for the different age group. This is the values for the patients in red and the controls in blue. It's not dependent upon age, except in some teenagers who spend the weekend not moving at all, like my daughter, for instance. And that's the longitudinal trend in the 95th centile stride velocity. Do you remember the arrows of PAT going like everywhere? The standard response measure of the 6-minute walking test leads you to conduct trials with 112 patients during 1 year. If you have something that declines much more continuously, then you can conduct trials with much less patients in a much shorter period of time. It means to decrease the burden of trials for patients. It means to accelerate the development of the drugs. It means to have an early signal of efficacy that allows you to know if you should or should not move forward with your drug. It means to avoid exposing patients to an undue placebo or to a drug that has side effects. And we know several examples from the last development in the field. So with with this technology, we could demonstrate in the application that rather than using 112 patients per group during 1 year with the 6-minute walking test, you could drop to 14 patients over 6 months per group because of the standard response measure. Then we were asked by the regulators if it's sensitive to change. The blue curve with the standard deviation are patients on steroids for at least 1 year and they decline. And the red curve are patients of the same age who are just given steroids. So this is the response to steroid in patients in comparison not with untreated patients, but patients who are already on steroids. So that's the boost effect of the steroids. And this boost effect of steroids, after 2 months or 3 months, you can quantify it. You can see after 3 months that the patient is doing better. So we were asked to define the minimally clinically important difference. We used a population-based approach and could find MCID of 10 centimeters per second. And 10 centimeters per second is basically 36 meters per 6 minutes, right? So about close to the 30 meters of the 6-minute walking test. Then we were asked to try to figure out what are the variables that may change, that may modify this variable. And we try to figure out if patients who are the best compliant, if the compliance of the patients influence the 95th centile start velocity. So if the patients who wear the device the longest period of time are the patients who are the best performer. And this is not true. The only factors that we could demonstrate to modify was basically the day of the week. You walk slower during the weekend than during the week. And the kids with Duchenne, but also the controls, walks a little bit slower during the weekend than during the week. We have to know that, and it means that when you instruct the patients how to use it, you have to pay attention to use with the same pattern of use across the trial and ask them to use it every day at baseline and during the trial. If they don't use every day, not to change the pattern of use. We could demonstrate that 180 hours allows you to have a 3% of variability. If you record only 50 hours, it goes up to 5 to 6%. So we propose that this measure should be performed during 50 and 180 hours to be considered as valid. This was a long and winding road, the discussion with the EMA. At first, it had to be a common assessment by the FDA and the EMA, but unfortunately, it could not happen. So it was a simple EMA assessment, and it started in June 2016 with the first discussion between us, hey guys, we should go to EMA. And the letter of intent that was sent at the end of 2016, and finally, the final endorsement by the CHMP was published on April 26, 2019. So altogether, more than 2 years and a half to get it. So we had a lot of interactions and feedback from the regulators. These are some questions and some notes that I've retrieved from the talk I had with the regulators at that time. And my personal feeling was that when we started this discussion at the EMA level like 3 years ago, we were like a little bit pioneering and discovering with the regulators this kind of new field, this kind of new devices, and it was very enriching. But we had to start since the very beginning, because the problem is that when you work for 10 years on something, you believe that everything is simple and clear and straightforward. This is not true. This is true for you because you are actually working for 10 years on that, but this is not true for someone else. So we discovered that sometimes you need to explain and explain and explain again, not because the person in front of you is stupid, but just because you are stupid. You do not appreciate that you know your stuff better than anybody, and that you need to be very, very clear and very simple to explain what you're doing. I hope I took the lesson on board, otherwise just tell me. So that's the list of questions we had. Another point is that it was important also to show that there is a huge support from the community, from the patients advocacy group at first, who understand, who appreciate it, that conducting trials with better outcomes allows to accelerate the development of clinical of drugs. It allows to decrease the number of patients on placebo, the duration of the trial, and unsuccessful trials. So it was supported by several applications and also several sponsors who took the device on board in different studies. So I understood it was also a strong point from, but maybe Francesca will tell you more about that, to be able to show that there was a huge support. So just to conclude, yes, it was a long and winding road. We discovered the process, and we discovered with the regulators this kind of new technology and how we should validate that. Then comes the new challenges. And the new challenges are Duchenne muscular dystrophy is a disease that is rare but not that rare. But we are conducting trials in very rare diseases. For instance, in disease in which there are 15 or 20 patients in Europe. It's just impossible to do it again and again and again and again for every single disease. And on top of that, we have developed outcomes for non-ambulant patients. But then it means that you have to develop outcomes for non-ambulant patients and for ambulant patients, and then to cross it by all the different diseases. If it takes 2.5 years for a single outcome versus a disease, you cannot conduct 20, 30, 40, 50 times the same process. So I think it's a real challenge. Another challenge is also to appreciate and to understand all the power of this kind of assessment. If with a single sensor you can capture any single movement of a patient during a long period of time, it means that you can not only extract a powerful outcome like the 95th centile stride velocity, but you can also extract several other outcomes that can be used according to the mechanism of action of the drug. Not only according to the disease, like the tremor, the dystonia in Parkinson, but also of the mechanism of action of the drug. Is the drug supposed to make the patient stronger, to increase the power? And then the stairs climbing is meaningful. Is the drug supposed to increase the endurance, the stamina? Then the walk perimeters is meaningful. Is the drug supposed to decrease to slow the disease evolution? Then the 95th centile stride velocity is more meaningful. So there are several outcomes that you can extract from a single sensor and try to tailor these outcomes not only to the disease but also to the mechanism of action of the drug that is supposed to act on this disease. And as Pat demonstrated, there are several approaches in Duchenne, several drugs that are used in Duchenne, but they have not the same mechanism, and anti-myostatin, for instance, are supposed to increase the power, and other drugs are not supposed to increase the power, but much more the endurance by working on the mitochondria. So that's what we have to appreciate also from this new technology, not to have a single outcomes for a single disease, but a kind of platform of outcomes that we can use according to the different clinical stage, according to the different drug mechanism, and to tailor our outcomes to make it tailored for the type of patients and for the mechanism of action of the drug. And finally, we have to solve the problem of the extension to other population, especially in the context of rare disease. Thank you very much for your attention.
Okay, so here is to show a bit of the learnings that we had on our side as an agency from this procedure. And I want, first of all, to thank Jane Mosley, who was the project manager who stuck through thick and thin with the consortium in the development. Well, as usual, as regulators, we don't have any conflicts of interests. So first of all, for those of you who are not familiar with the EMA qualification procedures, there are two possibilities. One is the qualification advice, which remains confidential and is not published in its content. And one is a qualification opinion, which was the case here, was a STRIDE 95th Centile. So the opinion is published in its entirety, is published publicly so that the public has an opportunity to comment. The questions are out there. And that's why I would like you to encourage to look at the STRIDE 95 Centile as a learning case, even if you're not interested in mobility, because it gives an insight, particularly with the questions, on the type of questions that the regulators had to the consortium and what was interesting for them to allow in looking at data provided in what was important to achieve the qualification. At the bottom, you have an aspirational timeline, which is 100 days or 130 days. This is the ideal timeline that we could achieve a qualification if we had no questions, but there were several rounds of questions here. Now, general consideration for digital mobility measures, Laurent has covered many of those. The fact that it is a continuous measure, it's interesting because it minimizes variability and event singularity, but what do you do when data are missing, when suddenly you see spikes of mobility or lack of mobility? So all of these considerations need to be part of what is prepared by an applicant that comes across to regulators for the validation. What do you do with a different type of data collection, with data that are outside a pattern? How do you interpret them? When do you decide to exclude them? How do you know that they are relevant or not relevant? The advice over a supervised test, and here Laurent has spoken extensively, it also can help distinguish between the performance, so what you can normally do, and the capacity, so what you can maximally do. And depending on the disease, this may be more important in some contexts. If properly validated, we expect that this could become a primary endpoint. At present, it's validated as a secondary for use. And as we have heard the day yesterday, but in Europe is particularly important due to our legal framework, data privacy and data protection need to be respected, and we expect applicants to set in a framework that allows this. Now, we touched on this yesterday as well. The impact of the device and the internet access has both the opportunity to involve patients who live far away, avoid the Tour Eiffel effect in certain cases, but also may end up in excluding part of the population that either doesn't have access to advice or, to the device, or that, for example, may be a risk, for example, of theft. Some people were in certain other advices were told that we don't want to wear something that looks like a Fitbit because we are at risk of being robbed, for example, in certain countries. So there are some considerations that may need to be thought in this respect. And one very important point that we haven't talked about much is that the development of this type of systems needs to be accompanied by a best practice. And this was a point that was discussed quite at length to try to understand how the patients are instructed for the optimum use, how the physiotherapists are instructed, how it works in practice, both in the clinical trial setting and when or if it needs to be utilized in the real world post-approval. So measuring time, in which environments, at school, every day, what times of the day, what training or support is offered, whether there is a feedback or monitoring of what the patients, what happens to the data, how compliance is assessed, and the feedback that the patients have. And this is, I know, we talked that we shouldn't burden the patients too much, but it's important that both the patients and the healthcare professionals are involved in the development of this best practice, because for the acceptability and the compliance, this is fundamental. Now, the first point we have covered, they need to be anchored, as we have seen, to some clinically recognized instrument, and the discussion when the applicant comes should discuss the limitations of the measure and the advantage or what new is brought by the digital technology development. The generalizability to other conditions is one point for discussion. Normally, of course, the context of use will need some form of validation also in different conditions when these come along, although probably not as extensive as we have seen here in the Duchenne. And also the symptoms, as Laurent was mentioning at the end of his talk, may vary depending on the disease. So in certain diseases, mobility of falls may be more important than in others. And so you need to discuss when you're trying to extend the use of a certain device to other population, what is relevant for that population. Learnings specifically from this procedure, and these are apparent from the CHMP questions in the documents I showed you before, is that, first of all, a detailed proposal for the context of use was requested, how they had to adjust for confounding covariates, and the impact of extending the duration of recording. So this Laurent has covered extensively in his talk. The distribution and the patterns of the data recording, the analysis and discrimination between different degrees of severity, the longitudinal correlation between the 6-minute walking test and the stride velocity, and the annexion of the best practice. I mean, this was really an important point to understand how the patients were using it. We were really fortunate to have a team on the side of the company and the consortium that was really dedicated and responsive. This made the procedure, although very long, successful. I mean, they were willing to be pioneers in this respect. They were responsive. They were prepared to discuss in details, formally and informally. We had several rounds of questions, and this went well thanks to that as well. Also, one other point was that we had access to non-public data from other trials. These were kept confidential, but this highlights one point that was raised yesterday. In the non-competitive space where certain data could be discussed, it's important in certain cases, and this for us was really a bonus. The applicant also supported the publication of the opinion, so this was completely transparent with the questions and what was put forward. Again, this is something that allows advancement of science and the regulatory science when we are in an area that is new. The patient involvement, they were involved throughout the discussion, and they were giving input from their own experiences and the best practice, I've said it many times. So I would encourage you really, whether you're interested in mobility or not, to look in detail at the document. There's a link at the end of this presentation, because there's quite a lot that can be learned on how the interaction went and what points were raised by CHMP. Thank you.
Thank you, Francesca. Thank you, everyone. We wait for Michelle for coming to, thank you, stage. And then we'll start a panel discussion. Thanks. Thank you, everyone, again. Michelle, maybe you want to introduce yourself since you're the one.
Good morning. My name is Michelle Campbell, and I am from the Office of Neuroscience, where I lead stakeholder engagement and clinical outcomes in CEDAR at the FDA.
Thank you. So a few points of reflections. The first one going back to the initial talk by Pat and really what has been talked about yesterday and even in closure here by Francesca, the importance of engaging the patient. We've all talked about that, like that's important, that's your customer to the measure you're building. Maybe the next step of the question would be how to do that. There are different ways, right? You can engage them up front and then maybe zero in a subcohort, or you can stay as wide as possible until later on. provocatively, you could always say, well, you engage it at the beginning and then you go your own way because you want to be disruptive in a way that the patient doesn't even know that they need the product that you're building. So what have we learned in this direction, and are this learning maybe generalizable to other use cases? Yeah?
Well, I think that patients' involvement since the very beginning is super important. And to a certain extent, in our experience, there are things we did well and things we did not. I, for instance, 2 patients with neuromuscular disease in my team. One with facioscapulohumeral dystrophy and the other one with spinal muscular atrophy. And they are both working on the project. So I think that at first, working with patients and welcoming patients in your team as engineers, as biologists, is super important. They have a lot of insights. So the guy in charge of the project in my team has a facioscapulohumeral dystrophy. The second thing is that the Duchenne community is quite a buoyant community, and there are several workshops and discussions in which patients' associations are always present. So there are formal and informal discussions about the early data, because the data were presented through the process since the very early data acquisition to this kind of workshop, and the discussion and the opinion of parents was taken on board. What we did not do well is that in the process of saying, okay, we don't mind about if the device is beautiful, we don't mind if the device is sexy, we want to have the right sensor calibrated the right way, and then at the late stage we will have something beautiful. It's intellectually understandable, but if you have to wear something that is ugly and that makes you look like a prisoner out of jail with an electronic brace, you can appreciate that it's 1 year over a process of 10 years. But on a daily basis, you have something ugly on your ankle and on your wrist. So this is something that was probably neglected, and that also at first decreased the compliance in the first recording, especially for the long-term compliance, and then also gives an image of something, okay, a gadget, but nothing more. And that's not easy, and I'm not saying that there is an easy solution. If you take this long way to create the device and to make the device exactly according to what you need to do, the exact context of use. And so the opinion of the patient was probably take a little bit too late in terms of designing the device. Today it's now taken fully on board, but probably it came quite late in the process.
Yeah.
So I'll make a plug for the patient community. I think that patients need to be invited and involved right off the bat, because otherwise you take a lot of years to create something that may not be acceptable by them in many respects. You also forget that in Duchenne muscular dystrophy, a lot of these young men have skin sensitivity. So if you're creating a device for them to wear and it feels wrong on their skin, they're not going to wear it, or they're going to take it off and put it on their dog if they can. So that will give you probably a different presentation than you anticipated. I also think that for the patient community, the reason that we have extended the years of life of these boys, the years of mobility, is because the patient community demanded the use of steroids in the United States. They pushed cardiologists in a corner and said, we want to start cardiac meds. And in fact, in Europe, because many of these boys, 60% are off their feet, when they were not included in trials, and it was very clear that we were concentrating on an ambulatory population, the international community got together and said, let's figure out how to measure those arms. And they developed the PUL, or the performance of the upper limb. They brought in physical therapists. They brought in all the people necessary to really think about what's important to these boys. And in parallel to that, we did preference studies to understand from those young men's point of view, once you lose ambulation, what are the most important things for you in terms of those arms, what they need to do, and getting your fingers on the table, putting your fingers to your mouth, being able to comb your hair, having privacy. So I think you have to understand that we're in this together. We're stakeholders as long as with everyone else. And if you don't invite us early and often and keep us informed along the way, then you sometimes have to take longer to get to your end, which is the end that's all important, right? So I think involving the patient community, asking them what you need, they fulfill it with money, preference studies, policy. They'll do anything they can to advance therapies in Duchenne.
So I'd like just to add that the agency is very supportive of the patient-focused drug development initiative and movement that has occurred over the last decade, I would say. And yesterday, Chris Leptak described some of the provisions under 21st Century, specifically to qualification. And another large piece of that passage was a patient-focused drug development section. And so if you're unfamiliar with that initiative, let me go back a little bit further and first describe what happened under PDUFA 5, which was where the agency was tasked with hosting what we call patient-focused drug development meetings, so we can learn from our patients, because we know that patients are the best person to tell us about their disease and their condition and the progression of it to the best of their abilities. And as well as their caregivers, in the example of Duchenne, also can provide a high level of insight. And during that time, those 5 years, the agency had to hold 20 meetings, and we actually held up to, as of right now, we've held 26. Which may not seem to a lot, but is a lot of work to determine. It was a task to determine what was needed. And we recognize that every disease is important, and we want to learn from all of them. Offshoot from that developed a nice offshoot of another way to engage with the agency was through some externally led PFDD meetings where patient groups could work with the agency to host things offside of the FDA campus. But this was very successful during that time. And as our next round of PDUFA negotiations happened, we were encouraged to continue to thrive this program and go to the next step, which was how do we really bring in the patient voice even more into our medical product development work. And one of these outcomes that has come from this initiative is a guided series on how to start from the very beginning of engaging patients and patient groups to all the way to how do we get to an endpoint. And it's a series, it's a very non-FDA traditional guidance series where the guidance series is written for our patient groups. It's obviously also written for our sponsors and other stakeholders, but it really is to help our patients know and understand how they can play a part and role in medical product development and collect the information, because we fully need to know what's most important to patients and how does that impact when we're looking at the data in the big picture. So I would say that patient involvement should be throughout development when applicable, and that we often look and see what was involved in that. So we would say that we should always be involving our patients at every step.
And one last comment, the patients are going to bring the payers in, because if you get an approval using a device, as we know, the payers, usually the first answer out of the payers, especially on an accelerated approval, is no. So the patients have to learn to navigate, have to learn to explain what value that outcome measure meant in terms of quality of life, et cetera. And so they're going to bring all stakeholders together so that once these therapies are approved, they're accessible for everyone.
Thank you.
Can I add a couple of points to this? Yes, I would say that it's also important to understand how to involve the patients, not only from the side of the company, which is fundamental at the stages of development. We always also involve them on the side of the regulators, on the HTAs, so the payers, when they come, because they offer different perspective. We found generally probably that for rare diseases, the patients are better prepared than in other therapeutic areas, maybe because they have more of a community to discuss what the needs are, they have a better understanding also of the regulatory process, and they have given us really valuable input. And there was also very valuable input in the public consultation, I think. So when the opinion went out, we got lay people feedback that was really important for us.
This is great, thank you, and I like it. A lot of these tips are very practical. Like, it's again a blueprint to do this again. And along these lines, we touched upon during the presentation and yesterday as well, some of these measures have an applicability that goes beyond the indication that we're approved for. So in this particular case here, we're looking at a measure of mobility in general that could apply to other conditions other than Duchenne. So my question would be, how do we reuse what's been already done and extend it to other indication, other therapeutic areas? Is there a regulatory pathway to do that, either in EMA, FDA, or both? And B, how do we extend it across the ocean? So this was, done at EMA. Is there if someone had to, redo the same application to FDA, would have to start from scratch, or they can reuse some part of it, and if so, which part?
Do you want me to start? Sorry, I don't have visual cues on that side. So yes. So how do we reuse it? So first of all, you have achieved validation as a secondary endpoint in a certain context of use here. And as both Laurent has said, and I said in my talk, for other diseases, there may be aspects who are more relevant and minimally significant changes that are more relevant for certain diseases and different, perhaps. So there would be required a degree of validation. So now you have proven that your system is valid in the Duchenne population of a ambulatory Duchenne population. So of course, if there were different degrees of severity, very prodromal populations, you would have to define at least what the minimally relevant change would be for those populations. If there are other symptoms, if falls are particularly important in another disease, then we would need to look at sensitivity for this type of aspects. So there is an element of looking at which population of use you're going to utilize your system and validate it, which is probably not as extensive as the initial validation is required.
So, Chris, do you want me to start, or do you want to start?
You're on the panel.
Oh. Well, I'll defer back to you. So it would be very similar. So obviously, what is the context of use? So what are we studying? What is the population? So when we are starting to expand, which of course, that's great if we can start expanding, movement hits many diseases we know. And so we would want to make sure that we can reliably, interpret the same way. What does the data look like? Is there any differences? So it really goes down to that interpretation. So additional data would be needed. It is that understanding of what is going on in the patient population. So, we would be very similar in terms of that. To the question of, well, if someone was coming to FDA for this, well, so this is we have accepted this into our qualification program. There is an accepted letter of intent. The question is an interesting one to respond to about the use of what can we take from past experiences. I think we always encourage, if we can learn from past experiences, to help the next submissions. And one of the nice pieces, if you are in the qualification program with our transparency provision, is you're able to see what our responses are and what those questions are to help, IQ as a way of how can we help learn from one another and enhance submissions that come in. And so I think if that scenario was to come, that would be a great question and dialogue to have with the agency about how can that be used in a way that is fruitful to any type of submission. Chris, did I miss anything?
No, I think ultimately both your response and Francesca's response at the FDA, we're not going to be agnostic to something we already know. So around the measurement, the analytical validation pieces, et cetera, to the extent that they apply, we're already familiar with that information, so the question might be a right of reference, depending on if it's the same party developing the information or not, if it's in fact proprietary or patented. That would be maybe a stickling point. It's hard to say. But in general, to expand into either a new population from a secondary to a primary, that lift for the incremental improvement is a much smaller lift than the initial engagement.
And just to follow on the discussion that was yesterday, it's always possible to have sorts of rolling or subsequent advices to expand on a nucleus of initial validation. And that's probably, like yesterday we were discussing, you start with a proof of concept and then you go on to full validation. In this case, you start in one population and then you would move on to others.
Thank you. You want to say something?
A little bit related to both of the last topics. Laurent, you told us about how the patients or the families train and kind of try to work on getting them their characteristics such that they would be in trial. I'm wondering, that is all part of kind of a feedback system. And in the drug development programs that I've been in, we wanted to provide no feedback. We wanted to be kind of completely objective, no feedback to the patients or their families as to how the drug was responding, and certainly having something on you all the time that tells you what your current strides are or whatever is feedback. And I wonder if it may not be an important thing for Duchenne's because maybe it is, but how did you include that in your thought process in terms of making sure that the data you were getting took that into account, took it away? Because I can imagine, we all wear Fitbits and what have you to make sure we continue to walk, right, or get up from our desk or whatever for not for disease, but for just kind of a reminder. Do the device that you're using, does that data get back to the patients and the family or not? Was it taken into account? How was it taken into? I'd kind of like to hear, because that's going to be maybe more important for somebody with a different disease, maybe not Duchenne, but maybe with sarcopenia, it ends up being a trainer, right, or something like that.
So, thank you for the question. We started from the philosophy. When you measure something, most of the time you change what you measure. And that's what happens in the clinical trials. If you perform 10 tests in a patient, I can tell you that he is exhausted. And that the test number 8, 9, and 10 will not be the same that if you had done it without test 1 to 7. This device was specifically conceived from a question that was clinical trial. We wanted to make clinical trials shorter, more efficient. So we thought only from a clinical trial perspective. So we pay attention not to change what we were supposed to measure. If you give feedback to patients, you change the measures. The best example being a Fitbit, an Apple Watch or whatever. You receive it for Christmas and then the day after you start running more hey guys, I did it. Since the very beginning, it was clear that the device and the way we developed it was not conceived to give a feedback to patients. Then you can imagine in future development as a companion device or whatever. But that was not initially taken on board of the project because the primary outcome, the primary objective of the project was to conduct better clinical trials, not to change the behavior of patients.
I have a question or comment. So this is Diane Stevenson from Critical Path Institute. So just some learnings. It was really insightful to hear the lessons learned from your experience in Duchenne's, and we led the first qualification opinion with EMA for the use of dopamine transporter imaging for use in Parkinson's trials, and I think Francesca was part of our initiative. And so we also had a long and winding journey, but some of the learnings we heard today, I think what we're hearing as a consortium is we want to find ways with remote technologies to have this journey be faster, right? And some of the things that you shared today are really important for us to realize as a community as we try to engage regulatory agencies on any disease where we're considering remote technologies around the responsiveness that you had in this experience to requests from the agency. That's typically takes us a long time, because when we're asked to bring in more data, that takes more time. And then your experience also is really rewarding to hear that you were able to have access to clinical trial data and confidentiality, right? So this is an area where the industry as a whole could help be very responsive. When we say data sharing, doesn't mean the whole world gets to see the data, but to inform the regulatory path, this is a key critical success factor. And then the other thing that I'm not sure how much was emphasized, but in your experience, the use of looking at the device in normal controls in a normal age-matched population was really important, and this is something we're dealing with now in embarking in the Parkinson's space. So I think these learnings, as all of us continue to think about how to accelerate the path forward for all the diseases we work on, the topic of, the rigor and looking at normal subjects, as well as being responsive and sharing relevant data from trials are the things that all of us can do to make this process faster and efficient with both regulatory agencies. Really good example case. Thank you.
So thank you very much. Indeed, I think that it is a very long and winding road, but because it was probably the first time that it was done, and everybody was learning a lot. And the outcome was positive. Also, there is something that is related to the size of the company and the consortium trying to bring it. I mean, Sysnav, who is the primary manufacturer, is a small company, so which means that if all of a sudden many other analyses are requested, there are not 10 engineers ready to jump on the topic and to do what is requested. So, I mean, it also changed a lot. When I'm at the SAG group, for instance, and I see a big company coming with 12 different people, and they are in London for in Amsterdam, for 2 or 3 days before, and training and rehearsing. And this is absolutely not the case when it comes to a small tech company where 2 or 3 people are just finishing the slides in the train to go to London. So it's really a matter also of the manpower that you can put on, because this is very time and energy consuming. The beauty was, as Francesca mentioned, also at the EMA, we had a point of contact that was very responsive, and there was a lot of good willingness from both sides. Regarding the controls, yes, it's super important. I mean, especially in children, because you cannot know what is related to maturation, what is related to growth. For instance, stride length is very dependent of growth, right? So if you measure patients during one year, they will grow. So you have the interference of maturation and growth. The other thing is that regarding the data from clinical trials, I completely agree with you. I think that whatever, if it's industry-sponsored or sometimes even non-industry sponsored trials, the fact that some consortium or industry believe that the patients wear a device or the patients conduct assessment, but it doesn't belong to anybody, it cannot be published, it cannot be discussed, it's a big problem. I mean, the patients, when they come to a natural history study, for instance, they don't get a treatment. They just do it because they want that the field evolve, they want to help, they want to support the move of the field. They don't want their data to be stored somewhere forever. I've never seen a patient telling, okay, I give my data for this natural history study and I want it not to be used or not to be discussed or not to be published. This is a complete nonsense. And so I think that it was indeed very positive to be able to use this data in the application.
Great, thank you so much.
Sorry, just also to add, coming back to the point about industry sponsors, I think it's important to note that, everything that Laurent presented today was actually there was non-investigational products, right? This was control arms, this was the steroid arm, which would be your standard of care. So you can have an industry sponsor share that data as a pre-competitive effort to develop the endpoint without actually revealing their investigational product. And so I think it is really important that industry sponsors see that they can be part of this without necessarily giving the game away or having to go through sort of non-disclosure processes.
So just concretely on this same theme that Diane brought up about the data sharing. So operationally, was the person power to enable the sharing, that was done, was that provided by the EMA staff, or was there a separate safe harbor space that managed this interaction? And this really gets to who do we ask to organize this? And does the agency do it and provide the manpower and do all the paperwork and communications necessary to get people to agree to do this? So who is doing that? Who's doing all the work to allow for this data sharing to occur in this example?
Okay. If I had heard properly, because there's a bit of echo in the room.
I'm sorry.
But, so who does the groundwork to allow the data sharing? That's the bottom line of the question. So we don't do primary data analysis at the EMA. We got the reports of the studies and the clinical summaries. So that was what was shared. And as Ieuan was saying, most of it was on non-investigational arms. And the company provided the data. So basically, this is done under the willingness and responsibility of the company wanting to share with the regulators.
Okay. So I mean, again, this just gets to, and Diane can comment on this, this gets to, in our data sharing space, just the person power and the processes to do this even when there's agreement. And one of the issues we run up against frequently is just literally getting all the agreements and things in place when we work through the foundation of the NIH at all. I mean, and so I'm just wondering if there are, should the FDA, should the FDA have an office that helps facilitate this?
So I will comment on that. And,
it'd be great to have a resource.
No, I completely agree. So there's a number of components to your question. First off, when information is collected in the IND space in an exploratory fashion, there's no obligation on the part of the sponsor to provide us with that data. So many times we don't even know that it exists. If there's an opportunity for us to look at it or to gather it, we would certainly welcome it. And around who's doing this, at least in the qualification space, the data is being compiled by the outside parties through whatever agreements they've established as part of their body. What we do, and Francesca mentioned this in her presentation, is that if we have data in-house relevant to the question, we will certainly use that. And we did that for total kidney volume, where we needed another data set for the validation of the model, and we happened to have one, so we used it. So there are ways that we can be creative in an appropriate way. And if there's an opportunity for us to get the information, like you said, in a safe harbor context, honestly, at least from the examples that I'm aware of, even when you create a safe harbor, there's no guarantee of success. And at least for the Critical Path Institute, they've had a biomarker data repository now for a number of years in the clinical safety space for organ toxicity. And through our efforts of contacting companies, encouraging their submission of the information, their efforts of doing their own outreach, although they've had some successes, the successes have been relatively small.
And I can tell you from the patient community, we went to the Critical Path Institute and started what we call the de-risk consortium, which is Duchenne specific, and we encourage, persuade, try to bribe, do whatever we can to get all data to go into that system so that we can advance the disease. So I think patient advocacy community is very much interested in making sure both negative and positive studies are shared so that we can learn from them.
I just want to put in a plug for qualification that one of the benefits, many benefits of the CARES legislation is that we do have a budget that helps to support some of our initiatives. So what we've done in the Section 3011 qualification space, if we used a good chunk of our budget that we've received each year to establish seed money grants for qualification efforts. So if you have an LOI that's been accepted into one of our qualification programs, you can apply to one of the grants. And the grants, they vary depending on how many grants and how much money we have in any given year, but they've been around $250,000. And so we did our first years of grants last year, and the announcement is out and will close in a couple of weeks. But think about it for next year if you're not currently engaging with us.
Just by way of update, I want to acknowledge this is a Duchenne-specific discussion, but the Critical Path Institute de-risk consortium that Pat just mentioned has been successful in acquiring over 5,000 individual subjects into our integrated database for Duchenne's, and is going through the formal fit-for-purpose path with FDA for model-informed drug development, and we're making great progress. And then we're so grateful, Michelle mentioned yesterday, to be achieving funding from FDA to start a new rare disease initiative, and we do this as part of our neutral role in bringing together diverse data sets across different diseases.
Is the grants available for non-US based institutions?
You have to have a US partner.
It's a US program.
So I'll leave it at that. We do have companies that are engaging with us that have European arms, but they qualify because they had a US sponsor.
Thank you. 5 more minutes. Okay.
Sorry, and this is not a question, this is as well maybe a part of an answer. So I'm from Sysnav, the company developing the device. And I think we've learned as well things along the way, and one of the thing that we're trying to implement now is to improve our contract with with the sponsors of the drugs we are developing and so on, to put clause in them that allows us to use, either the placebo arms, either like all of their data into, the qualification documents. So of course, would not be allowed to publish. And then, like retrospectively for like studies that were there before, is trying to have a negotiation with them and so on, right? It's not that easy, it's not always easy and so on, but I think this is essentially like a lot of the work that we have to put into, preliminary to putting the package together, which is again like another part of the job. But like just for all the others, outcome developers here, I think this was like sort of one critical piece that we are missing at the beginning of like what is the data sharing agreement, how like where do we draw the boundaries, what can we like have and so on. Having this discussion up front is very complicated with big pharmaceutical companies, but this is like for us this is a sort of a need that we would not. I mean, this is a must essentially. Like we cannot take any other route, right?
Thank you.
And perhaps just as one further small comment, we also welcome disease progression models based, for example, on registries or other diverse data sets. So these are also been evaluated in other procedures.
So I have a question related to, and I appreciate the incentive to we're trying to create an evidence-based framework. And from a patient perspective, I'm going back to the timeline. So one incentive would be a grant, right, to after the LOI, but there was 4 years between the LOI and the EMA acceptance, correct? Is it going to take 4 years here to get this through the FDA? Because that just doesn't seem, rational. If we're trying to use new technology to adapt for patients, so I don't mean to put pressure on anybody, but if we're going to develop a framework, maybe we should look at evidence.
Just a very small comment. Subsequent qualifications haven't taken that long. I think that, the first ones always break a lot of walls and new ground, so things are improving.
I mean, Francesca mentioned as part of their qualification effort, part of it is based on the quality of the submission and the questions that need to be brought forward. If you get all your questions asked in your original submission, the review time is not that long. Science doesn't work that way usually. Although the cures process brought many benefits, one of the downsides is that we are now a formalized three-step process. So we have to go through all 3 steps according to the statute. We will do what we can to make those steps progress rapidly from the one to the next, but we can't skip steps. So that's where we're at.
And to go back to a message that you were mentioning yesterday, we cannot underemphasize the importance of coming in early, because we can help you to frame your questions properly, to discuss informally what we expect to see, so that when the application comes, it's expedited.
But I'd also like to add that there are other ways you can work with the agency early on to try to make sure you're headed down that right path before you submit a letter of intent to a qualification program, and making sure your submission will have the appropriate information. So one of the initiatives is of course our critical path innovation meetings, which has been highly successful in CEDAR, and as well as the other medical product centers, CBER and CDRH, have some type of mechanism. It's termed differently in each center, but ways to engage early with the agency to get some high level thoughts about what's going on and things like that. So we understand with that time, we get it, we feel the same way a lot of times, but really, if you are early and you're thinking this is something that is relevant or what's going on, encouraging some of the other ways to engage early could be useful.
We're out of time.
Yeah, so I think we're out of time. This has been a great discussion. It's been really helpful. Great panel. Thank you very much. So, and give them a,